carbohydrates today. So that's sort of my plan. We're examining carbohydrates from a structural perspective. Later in the term, we will look at them from a metabolic perspective. And I think from a metabolic perspective, you will get a much keener insight into the needs and the considerations your body has for energy. And that will be coming, as I said, later in the term. For right now, though, we're talking about structural considerations of carbohydrates, and I want to pick up where I left off last time. So last time I showed you some cyclization that happened for the furanoses and the pyranoses based on the fact they were named in those molecules that look like that. You saw glucose that made this ring structure. I pointed out it could exist in two different forms. And that the critical thing was that this was a reversible reaction, and so long as that anomeric hydroxyl was intact, that is unaltered, then that reverse can happen. Once I alter that, then I lock it into place. When I alter that anomeric hydroxyl, I create what's known as a glycoside. And we'll say a lot more about glycosides later. Similarly, fructose makes a ring structure. You can see it has 5 members. And as I pointed out last time, both glucose and fructose actually can do both 5 and 6 member rings. You're only responsible for glucose as a pyranose and for fructose as a furanose. Okay? So that cyclization in both cases involves the carbon with a double bonded oxygen. So in the case of fructose, it's the ketone group. In the case of glucose, it's the aldose group. And that is always the case. That always is involved in the cyclization. Okay, now, there's a figure there. You see that this just simply shows you that fructose can exist in a 6-membered ring. You're not responsible for the 6-membered rings, okay, for fructose. All right? You are for glucose, however. All right. Well, there are other considerations, it turns out, relative to structure. Last time somebody asked me about, well, whether it was the beta form was more stable than the alpha form or vice versa, and I said the alpha form actually was a little bit more preferred than the beta form. Part of that is due to steric hindrance, but of course beta form does exist. And so when we look at the beta form, we start thinking about, well, are there steric considerations that we have to think about that might affect this molecule? And it turns out that in fact there are. So that hydroxyl sticking up there can interact with that CH2OH group that's also sticking up there. And so that causes some actual adjustments that happen inside the molecule. Now the adjustments I'm talking about here are not flipping OH groups. The adjustments are actually shifts or slight twists in the molecule that cause it to adopt a different shape. Okay? So that we're not flipping OH groups. They're not going alpha, beta, or doing anything like that. We're simply changing the shape of the molecule. And this molecule, in this particular case, there are 2 predominant forms that we see for a 6-carbon ring, just like this. And they're known as the boat and the chair form. Okay? So the chair form is one way of twisting. When we do that, we see this OH, which was pointing upwards, as over here, no longer is pointing upwards, it's pointing away. And the chair form is a sterically favored form compared to the boat. So the 2 possible forms that can exist for a given beta in this case are the chair and the boat form. And the boat form does, in fact, form. So it's not like it's an absolute thing. But of the 2, the chair form is much preferred just because it's avoiding a lot of that steric hindrance. And 6-carbon sugars in general can adopt boat and chair forms. So this is not something that's unique to glucose or anything like that. So the twisting of the bonds facilitates either of these 2 configurations that you see here. Similar things exist for 5-membered rings, but they're a little harder to visualize. Okay? And to be honest, I'm not going to hold you responsible for understanding the 5-membered ring configurations. I don't think that it really teaches us anything instructive. The boat and the chair is something that you can envision in your head. A chair shaped like a chaise lounge. A boat like a chaise lounge with both of the things sitting up would be very uncomfortable. A boat sitting like that would be very uncomfortable to sit in something like that. This to me looks more like an envelope. Okay, so there's the base, and there's the top of the envelope. But I'm not sure what you call that, a sideways envelope? I don't know. So, the Postal Service, maybe they have a term for that. Okay, so that's what I want to say about the simple the simple monosaccharides, the simple sugars. Okay. Now I want to turn our attention to some modified sugars, and they're not overly complicated, but there are some things that can happen to sugar molecules, and you should be aware of them. Okay. So first of all, there is some chemistry that's a consideration for sugars. All right, sugars can, for example, be oxidized. Okay? They can be oxidized. The most commonly oxidized sugars are the aldoses, because the aldehyde group is relatively easily oxidized to an acid group. So what you see on the screen is a glucose that is being oxidized to an acid form. When we talk about oxidation, we talk about loss of electrons. And whenever we have loss of electrons, something else has to gain those electrons. That is, for every oxidation, we have to have an equal and opposite reduction. So the oxidation, glucose is becoming oxidized to the acid form here. Something is getting reduced. Those electrons, in this case, are going onto a cupric ion, copper plus 2, and reducing it to a cuprous, copper plus 1. Now that reaction actually involves a change color, all right? A color change. Change color? A color change, okay? So that makes this reaction very easy to study. And we're not going to worry about what the colors are or any of that, but it's a very easy reaction to study. Glucose, as a consequence, is referred to as a reducing sugar. It's called a reducing sugar because it can cause something else to be reduced. In this case, copper plus 2 is becoming reduced to copper plus 1. And in the process, glucose is becoming oxidized. Now, ketones are not nearly so readily oxidized. Ketones like fructose are not nearly as strong of a reducing sugar as glucose. In fact, for our purposes, we will essentially consider them as non-reducing sugars, although you can get fructose to oxidize a little bit. Okay? So, ketoses do not oxidize nearly as readily as do the aldoses. Notice, notice here that the oxidation requires an aldehyde, right? Well, if I have a ring structure, do I have an aldehyde? No, I don't. So if I have a ring structure, I can't oxidize a ring structure. I have to have straight chain in order to oxidize it, which means that I can't have a glycoside. Can't have a glycoside. That is, if I alter this OH group, it can't go to the linear form, and if it can't go to the linear form, I don't have an aldehyde. Therefore, this reaction will not go. So a glycoside will not be a reducing sugar. Okay. Here are some glycosides. I've described them to you in words. Now you can see them in pictures. Here's a glycoside in which the hydrogen of the anomeric hydroxyl group has been replaced by a methyl. Okay. This is it's called a glycosidic bond. It's called an O-glycosidic bond because the oxygen is still there. We'll see later there are also N-glycosidic bonds in which the oxygen is replaced by a nitrogen. Okay? But the fact that we no longer have a hydroxyl there tells us that we have a glycoside. And in this case, it's an O-glycoside because the oxygen is still present. You see it in the alpha configuration, you see it in the beta configuration, but neither one of these can swap into the other because neither one can go back to the linear form that we saw that was possible when we had the hydroxyl on the anomeric group. Okay. Here are some N-glycosides, and as I said, N-glycosides are sugars in which the OH has been completely replaced, in this case by an N with something else attached to it. That is known as an N-glycosidic bond. And we'll see, again, we'll see some examples of that later. Yes?
Why can't we go back to the whole form and not have an OH anymore?
Well, it's rooted in the fact that it can't get back to aldehyde. So that once you put that methyl on there, it's no longer capable of flipping back to that aldehyde or ketone group. So that's the nature of a hemiacetal. So it can't go back to that. I'm sorry? Well, chemically it can't. So a hemiacetal, you should go back and review your organic chemistry a little bit. A hemiacetal cannot reconfigure in that way. A hemiketal can't do that either. Okay? Other questions? Not too many people here today. Everybody decided to take off today. Well, I don't know, maybe. Attendance is not required, but at the same time, in my experience, those who come to class benefit. So maybe just by being here, you guys are getting smarter, right? So maybe you're already benefiting from that extra credit. Okay, so answer to one of the questions on the midterm, okay? The answer to one of the questions on the midterm will probably be glucose, right? So, will that help?
Yeah.
Okay. All right, so now you know you've got an insight to the exam. Okay, so there are a variety of modified sugars that we see. As I said earlier when I talked about carbohydrates, I said the simple ones had the CXH2OX configuration. These guys are examples of ones that don't have that configuration. For example, Fructose. If we look at Fructose, it doesn't fit into that configuration, that structure, because instead of having a CH2OH, it has a methyl group back here. And another thing that students frequently ask me is, since only a few ask, because once I told you you weren't responsible for L sugars in the ring structure, nobody thinks about them anymore, but a few people do. So for those few of you who think about that, this is what an L sugar looks like. For an L sugar in the ring structure, instead of going up, the group goes down. Okay? And that's why that's called L-fucose, because this last group back here is going down instead of being pointed up. So that's, but again, you're not responsible for that. These guys right here, acetylgalactosamine, acetylglucosamine, are found frequently when they are linked in glycoproteins, and I'll talk a little bit more about those today. It turns out that this guy right here, it's called N-acetylglucosamine, is a monomer of a polymer that's found in insects known as chitin. So when you've heard of chitin, it's actually a polymer of this material right here. And we'll talk about polymers a little bit later. Sialic acid is found on the surface of many cells. It participates in reactions involving signaling and also in the entry of things into cells. And again, I'll say a little bit more about those later as well. The last thing I'll talk about here are phosphorylated sugars. So phosphate, we've already talked about with respect to going on to proteins. Phosphate is also something that's frequently put onto sugars. So on the left, you see glucose 6-phosphate. Glucose 6-phosphate is a very common intermediate that we see inside of cells. As we will see later, this molecule is produced in the very first reaction in which we're breaking down glucose. So the very first reaction that cells use to break down glucose involves putting a phosphate onto it. Putting phosphates onto things takes some energy because you're making a molecule that has a higher energy than the one without it, at least in the case of sugars. Okay? So these guys all have higher energies than the same corresponding molecules without those phosphates. And that can be a factor in how they're made. That can also be a factor in their how they're broken down or how they're used. And we'll say a lot more about that when we talk about the breakdown of glucose. These three molecules here are all found in the breakdown of glucose. So they're all part of the process that we call glycolysis. Okay, disaccharides. Now, your book has one of the stupidest structures for sucrose that I think that it could possibly draw. All right? So what is sucrose? Sucrose is also known as table sugar. It is a disaccharide, meaning it contains two sugar units. One of these sugar units is a glucose, and one of the sugar units is a fructose. Okay? Now, this structure would be fine except for the fact that they've been lazy and they haven't really drawn it the way that it looks. Notice how they make this odd bond right here. And the reason they do it is they're trying to save space. Okay? Now that space might take up that much more room in their textbook. Okay? Because in reality, this guy is underneath the glucose. This is fructose. That's glucose. Okay? And they have drawn this so that it's sort of the upside down structure. In other words, look at this. There's carbon number 1, 2, 3, 4, 5, 6. We always put carbon number 1 over here on the right. Look what they've done. There's carbon number 1 on the left, 2, 3, 4, 5, 6. It's completely upside down. Don't memorize the structure. I have a much better one that I will give to you that's much easier to memorize. Sucrose is one of the molecules you need to know the structure of. Okay? Lactose. Lactose is also a disaccharide. It's a very common disaccharide. It's also known as milk sugar because it is the most common sugar found in milk. Okay? Lactose is comprised of one molecule of galactose and one molecule of glucose. Do you have to know this big hairy name? No. Do you have to know this big hairy structure? No. Okay? But you should know what is in lactose. Same thing of maltose. And you don't have to know the name of sucrose either. I mean, sucrose is sucrose. To memorize that name is kind of like a waste of your time. All right? But you are responsible for the structure of sucrose. You're not responsible for the structure of maltose. Maltose is comprised of two molecules of glucose. Now, I will point out linkages here to you because they become relevant, especially as we come and talk about polysaccharides. All right? The linkages between the sugars are critical. Okay? And we'll see that especially with respect to polysaccharides. This guy right here is in what's called a beta 1,4 configuration. Beta 1,4. The beta refers to the fact that the anomeric carbon is pointing upwards. But what about the 4? Well, since it is over here a glucose, the 4 always has a hydroxyl down. There's no alpha and beta on anything except the anomeric carbon. All right? So beta 1,4 tells us how the two sugars are joined to each other. In the case of sucrose, what we see is we have glucose in the alpha configuration, and we have fructose in the beta configuration. Now that should tell you there's something different about this guy compared to this guy. What's the something different? There's a big chemical difference, for example, between the two. And it's not the fact that one of them has fructose either. A big chemical difference. What's that? Bonded to different carbons. They are bonded to different carbons, but that's not the full answer. What's that? Intermolecular forces? No, not really. Yes? What about the rings? Well, but that's because this one's a fructose, so it's not that. No. Yes, sir? Ah, very good. The one on the right has a free anomeric carbon. This guy does not have a free anomeric carbon. So what would you expect this guy would do? This guy would be a reducing sugar, right? Because this would become an aldehyde, and that could then become oxidized. This guy, they're both tied up. It's what I refer to as a diglycoside. Each of those has a glycosidic bond to the other one. This guy can't go linear. Sucrose is not a reducing sugar. Okay? So, that difference of linkage really gives sucrose a different chemical property that, for example, than lactose. How about maltose? Where does maltose fit in the scheme? Reducing or not reducing? Reducing, right? For the same reason this guy is up here. There's a free anomeric carbon, and as a consequence, this guy can go linear, make an aldose, and we're set. Okay? Everybody clear on that? No? All right. Now, this is a high-quality art drawing I had commissioned to show you how sucrose actually looks. All right? Here's fructose, here's glucose. Now you see how much more space it would have taken the book if they put the fructose underneath the glucose the way it actually exists? All right? It wasn't like text, I mean, that you weren't paying enough for this textbook already. They couldn't have spared this much more space to do it. Okay? Textbook manufacturers are rips, absolute rips. Okay. Or rippers, maybe I should say. All right, so that's what sucrose, and that's, if you're going to memorize the structure, there's a structure that I think you should memorize. So on the book drawing, which is back here, say it again.
Yeah, so you only got the corners going towards the other. Is that another carbon right there?
This right here? No, that's just, they're taking artistic license to make the thing fit the way they want to make it fit. That's not a carbon. Okay? So, yeah, I mean, it's really a stupid figure. Okay, and these aren't carbons here either, by the way. So these are just bends. They're fitting it in. At least in the case of lactose, that's a very commonly drawn structure like that. And the way it actually would exist would be you would have here on the right the glucose, and the galactose would simply sit like that. So they won't sit on top of each other like this guy does, but they would sit more like this. And that's really all they're trying to depict. Okay. Let's see. The awesome art? Yes, I will. I paid a lot for that, so I want to make sure that I get my money's worth out of it. There's the awesome art. Okay? Everybody got it? There's mud. So basically, glucose is in the alpha, and fructose is in the beta. That's what's up there. What was that? I'm sorry, I can't hear you. Da Vinci's work? Yeah. Leonardo da Vinci started with these, actually. He later expanded into other things, so I may have a career. You never know. I can't sing, so I should be able to paint or something, right? Mona Lisa. The Mona Lisa of Sucrose Lisa or something like this, yeah. Oh, boy. Okay, so let us move on. Polysaccharides. So polysaccharides, as their name implies, are polymers of saccharides. They have many sugar molecules within them. All right? So I'm going to talk about a couple of examples. The first one I'll talk about is cellulose, one of the most abundant polysaccharides on the face of the earth. It is a polymer of glucose, and it's a polymer of glucose that is the structural component of plant cell walls, or an important structural component of plant cell walls. Okay? It is, as I said, a polymer of glucose, and the linkage between individual glucose units is beta 1,4. Okay? So we have a beta 1,4. There's the up right there going from the carbon number 1 and 1,4, 1, and they've actually, let's see, what do we have here? They've actually flipped, they've twisted the molecule. So it's a little hard to see in that configuration. But the important thing is that it is a polymer of beta 1,4 glucose units to each other. And that differs from another plant polysaccharide that exists known as amylose. Okay? Amylose is a plant polysaccharide that exists as alpha 1,4 linkages. Okay? So alpha 1,4 linkages are different from beta 1,4 linkages. Now you see the word starch up here. Starch is not a specific polysaccharide. Starch is a mixture of polysaccharides. So if we're talking about something that has only alpha 1,4 linkages, we're talking about amylose. Right? Glycogen I'll explain in a minute. It's related, but you'll see why it's not exactly the same thing. Now, the difference between amylose and cellulose is that we cannot digest cellulose. We do not have in our bodies as human beings the enzymes necessary to break this down. It's because of this that plant material that we eat is roughage. That is, it sort of doesn't digest and gets carried through the system as it is. Okay. To break down cellulose requires an enzyme known as cellulase. And cellulase is found in the rumen of organisms known as ruminants, produced by a bacterium. Okay? Because ruminants have cellulase, they can digest the cellulose that's found in plant cell walls, and that's why you see cows going out and eating grass, because they can digest it. Now your dog eats grass, it can't digest the grass, that's why your dog pukes. Dogs use grass as a puking device. Okay? You have a dog?
Yeah.
And does it eat grass?
Yeah, and
Does
it pukes.
And it pukes. Okay?
They do it on purpose.
They do. They actually do it on purpose. They do. Yeah. Because they're dumb animals. Okay? So, no, I love dogs. Don't get me wrong. I really love dogs. But that eating the grass thing is not one of my favorite things that they do. Okay? So ruminants have this bacterium that makes this. And people say, well, what if we had the bacterium? And the answer is, it wouldn't do us any good, because the bacterium requires a very specific environment that's found in the rumen. It prefers an anaerobic environment that's found in the rumen that we don't have in our system. So if we had the bacterium, it wouldn't do us any good. But what if we put the gene into bacteria that we have, or what if we put the gene into human beings? What do you guys think about that? Let's do it. You want to be the first? So it would be an interesting experiment. Okay, it would be maybe what we should start with is dogs. You see, it would be an interesting experiment. I think you'd have to put it in the dog. See, does the dog go out and the dog gets frustrated? It eats his grass. It used to be able to puke. Now it can't puke. You know, you'd have to provide other means for the dog. I think. Okay, getting silly here. Now, on the other hand, we can break down amylose very readily. We have the enzymes to do it. Starch, of course, is an important component of many foods, including most commonly potatoes. And starch, I said, is a mixture of polymers. So one of the polymers is amylose, which is a polymer of alpha-1,4 linked glucose molecules. Okay. The other thing that we find in starch is a plant polysaccharide known as amylopectin. A-M-Y-L-O-P-E-C-T-I-N. Amylopectin. Now, amylopectin is also a polymer of glucose. It also has alpha-1,4 links. But in addition to having alpha-1,4 links, occasionally it has alpha-1,6 links. These provide what we call branches in the polymer. Okay? Branches in the polymer. Glycogen, which is a human polysaccharide, is related to amylopectin. It also has alpha-1,6 branches. So we can think of running all the way along here from right to left, left to right, a polymer of glucose, alpha-1,4, 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 alpha-1,4. Okay? Up here, every now and then we have one of these 1,6s, and it's a branch. Then this goes to the left, alpha-1,4, alpha-1,4, alpha-1,4. So we only see the 1,6 at the place where it branches. Why do we have branching? Well, I'll save that for later, because it turns out that branching is a very important property of these polysaccharides, especially as they relate to animals. Okay? Especially as they relate to animals. Glucose is much more branched than amylopectin is. Amylopectin has branches about every 30 to 50 residues. That is, about every 30 to 50 glucoses we see a branch. In glycogen, we see it about every 10. Glycogen is much more branched than amylopectin is. And as I said, it's actually rooted in the needs of animals compared to the needs of plants. So I'm not going to, as I said, I'm not going to tell you what that is right now, but I will talk about it later when I talk about glycogen and how we break it down. It's related to how we break it down, actually. Okay. Let's see. What else did I want to say here? That might provide a good place for a song. Would you guys like a song? Oh, yes. Okay. So, what's that? I just couldn't hear what you said. Oh, okay. All right. So this is the easiest song to sing that I've ever written. It's very simple. It's to the tune of "Hark the Herald Angels Sing," and it's known as "Hark the Sucrose." Okay? Please join me. Carbohydrates all should sing, glory to the Hayworth ring. Anomeric carbons hide when they're in a glycoside. Glucopyranose is there in the boat or in the chair. Alpha, beta, D, and L. Diastereomer hell. Alpha, beta, D, and L. Diastereomer hell. All right. Now, the song actually reminded me, I haven't told you what a Hayworth ring is. You guys can figure, what's a Hayworth ring? So the ring structures are called Hayworth rings. That's a name that you should know. Okay? And the straight chain structures are called Fischer structures. F-I-S-C-H-E-R. So Fischer, linear chain versus Hayworth rings. Those are terms that you will hear and you should know. Okay, now, that's the only song I have for today, so we're going to have to get back into this stuff now. Now, you saw modifications that happened to the monomers, such as you saw phosphorylation of glucose. You saw other things that can happen to those monomers. It's not surprising that we will also have polymers that can be modified as well. One class of those polymers are known as Glycosaminoglycans. Okay? I'm not going to spell that since it's on the previous thing, but Glycosaminoglycans are interesting modified sugar-containing polymers. They're interesting modified sugar-containing polymers. A few examples are shown on the screen. And what they are polymers of modified sugars, and the repeating unit is a disaccharide. So the repeating unit is, in this case, these two, in this case, these two, etc., etc. Okay? Now, look at the properties of these guys. Here's a COO minus. That was a COOH that ionized. There's a sulfate. Glycosaminoglycans, if you look at all of these, you will see that the modifications that they have all resulted, with the possible exception of that, all resulted in the formation of negative charges. So we could imagine that if we take and we make a polymer of these negative charges, we will make something that's polyanionic, and it's going to be a long chain of negative charges, right? A long chain, many, many negative charges. This causes these molecules to have some very interesting properties, okay? Some very interesting properties. They tend to be slimy, slippery when they're dissolved in water. They dissolve in water readily, and they're slimy, slippery things. Your snot, for example, contains some glycosaminoglycan-like compounds. Okay? Synovial fluid, which is the lubricating fluid that's in every joint of your body, contains a lot of this guy right here, hyaluronic acid or hyaluronate, same thing. Okay? So this long polymer with this long set of negative charges causes the solutions that they're in to have very different, very unusual properties. As I said, they become very slippery, and they do very good lubricating purposes. That's what you want in the oil in your car, for example, is that it is slippery. Okay? In the joints of your body, hyaluronic acid is very important in that respect. I'll also point you up here to heparin. Now, heparin is an interesting one of these molecules because heparin, look at the negative charges. 1, 2, 3, 4 on every repeating unit. This guy is really negatively charged. Okay? Really negatively charged. Heparin in the body helps to prevent blood clotting from happening. Its full function of the body isn't completely known, but it's known that it will prevent blood clotting from happening. If a surgeon, for example, wants to prevent blood clotting from happening during a surgery, they will inject heparin to reduce the incidence of blood clotting. Based on what I told you last time, I hope you would have some good ideas about why heparin might be a good anti-clotting agent. Any thoughts? Yeah?
Maybe it
Maybe it binds to calcium. Yeah, and since calcium is necessary for prothrombin to bind and recognize the place to start, if the calcium gets all mopped up, guess what's going to happen? Prothrombin is not going to have a place to dock and do its thing. Okay? So that's probably the reason why heparin helps to reduce the level of blood clotting. Okay. Now, what I just showed you are polymers of sugars, right? If I take those polymers of sugars and I link them to a protein, I create a protein that has a very interesting property, number one, and I create a molecule that has a new name. It's called a proteoglycan. Okay? A proteoglycan. Here's a schematic showing, okay, a protein backbone that has a bunch of different polymers attached to it. Now I want you to think about this structure for a minute. Okay? And based on what I told you about the chemical properties of the glycosaminoglycans, what do you suppose is going to be going on out here where all these things are now being sort of held in the same place? What do you suppose they're going to do? They're going to repel each other. All those negative charges are going to repel each other. Now I want to show you a proteoglycan. Look at that. They're all trying to get as far away from all that negative charge as they can. Okay? That's really something that the chemistry that you know about it confirms now what you actually see when you look at these molecules. Okay, so that's the polyanionic substances that I want to talk about. Now I want to talk about, it's sort of an odd thing at this point, but it'll become important for us later. And this is what's called a saccharide hybrid. So everything so far I've been talking about were monomers of sugars, polymers of sugars, or polymers of sugars that I attached to a protein. Now I'm going to attach a sugar to something else. And the something else I'm going to attach it to is a nucleotide known as UDP, uridine diphosphate. So UDP, of course, is, uridine is, of course, the base that's found in RNA. And it starts out as UTP, uridine triphosphate. RNA polymerase uses that to make the RNA. If we take a phosphate off of uridine triphosphate, we make uridine diphosphate. And if we link that diphosphate to glucose, we make something called UDP glucose. Now, this is UDP glucose right here. Okay? And the reason I show it to you right now is you will see this molecule later in the term. And my only reason for bringing it up right now is it's an example of a class of molecules that we'll talk about both this term and next term, known as activated intermediates. Activated intermediates. I'm going to give you a definition for that in just a second. An activated intermediate is a molecule, okay, that has a high energy bond in it. And that high energy bond is used to donate a part of itself to something else. All right? I'll give you the definition, then I'll tell you an example in a second. So an activated intermediate is a molecule that has a high energy bond, and that bond is actually right here, right there. The high energy bond is used to donate a part of itself to something else. Now, UDP glucose is used to donate a glucose during the process of making glycogen. So the part of itself that it's donating, in this case, is glucose. And that's necessary for making glycogen. Okay? You'll see more about that reaction later. But I wanted to introduce the term activated intermediate to you because we're going to see, probably over the next two terms, 3 or 4 examples of activated intermediates where they have that high energy bond and that high energy is used to donate a part of itself to something else. Okay. Now, again, this is a day for terms. The next term I'm going to introduce to you is a glycoprotein. This is one you've probably heard of before. And glycoproteins are different from proteoglycans. They sound similar, but they're different. Remember that a proteoglycan contains the long polymers, okay, known as glycosaminoglycans. A glycoprotein is a protein that has a few carbohydrates attached to it. A few. I won't define what that number is. You'll see some examples in a minute. But a glycoprotein only has a few carbohydrates attached to it. They're not polyanionic. In some cases, they may have a phosphate or two, but they're not anything like the glycosaminoglycans are. So glycoproteins are different from proteoglycans. Glycoproteins do many things inside of cells, but one of the examples I want to give you is that of making antigens. Okay? In your blood, you hear about people who have different blood types. And the different blood types arise from the fact that on the surface of blood cells, your body has produced specific glycoproteins. Okay? This is contained in your genes, which one you're going to produce. If you have type O, you produce a type O antigen. If you produce type A, you produce this. If you produce type B, you produce this. Now, we see a schematic representation up here. We can actually see abbreviations for what these things are here. That's galactose. This is N-acetyl glucose. This is galactose. And that's the sugar fructose that you saw earlier. Okay? The only difference between these three forms is the decoration that we see of this structure. This structure is common to all of these. That is, the left part is all in all three of these structures. A person who has type A blood is capable of putting that onto itself. A person that has type B is capable of putting that onto there. Okay? So the ABO blood types arise from different glycoproteins that are found on the surface of blood cells. You have an immune system that recognizes and says, whoa, I see the wrong type of thing here, I'm going to attack it. Okay? So that's why you don't want to mix bloods that don't have appropriate types for each other. That's a problem. And one of the things we see with glycoproteins is they're frequently used just for the purpose I described to you here. That is, they have an identity marker. Just like your student ID tag flags you, so too do glycoproteins on the surface of cells flag the cells. They may say, hey, I'm a liver cell. Hey, I'm Kevin Ahern's blood cell. Okay? Hey, I'm a kidney cell. Hey, I'm Kevin Ahern's kidney cell. That becomes important when we do transplants. Okay? If we transplant a kidney from one person that has one type of antigen on its surface into a person that has an incompatible setup, we're much more likely to have rejection occurring of that kidney because the immune system says, whoa, doesn't belong here, get it out of here. And it will take it out of there. Okay? So glycoproteins are very important in that identity process. When we look at the glycoproteins, we discover that the link between the carbohydrate and the protein occurs in two different ways. They're called N-linked and O-linked. Okay? The N-linked are made by linking, all right, in this case we see N-acetyl, okay, this is glucosamine, all right, so there's the N-acetyl part right here. Here's the glucosamine right there. Is this a glycoside or not? Yes, it is, because that's the anomeric carbon right there. That is an NH instead of an OH. So it's N-linked because it has an N there. So N-linked N-acetyl glucosamine, it's linked to the side chain of asparagine. So what's called N-linked glycoproteins have N-acetyl glucosamine linked to the to the nitrogen on the side chain of asparagine. Okay, the other type of linkage that we see in glycoproteins are the so-called O-linked glycoproteins. And this is depicted here. You're not going to have to draw these structures. Don't worry about drawing these structures. But you should know what's involved in an N-linked glycoprotein. An O-linked glycoprotein, on the other hand, links an N-acetyl galactose, that's what this guy is here, to the side chain of serine. Is this a glycoside? Yes. Okay. We replaced the H off of there. We no longer have an H. This is now a glycoside. Now, the ways in which the N-linked and the O-linked are made are slightly different. All right? They're slightly different. N-linked oligosaccharides, first of all, have a common core among them. So here is an N-linked glycoprotein. And the common core looks like this. There's our N-acetyl glucose linked to the asparagine. And we see this grayed out region right here. This grayed out region is common to all of the N-linked glycoproteins. This part is common to all of them. You'll notice there's parts up here that aren't in gray, and that means that these setups are what I call decorated differently. This is part of the identity. Okay? So different decorations correspond to different glycoproteins and actually tell different things, as we shall see. I'm not going to talk about it today, but I'll talk about it next time. We see different decorations out here tell the cells. For example, this guy needs to go to the Golgi apparatus. This guy needs to go and get exported out of the cell. This guy needs to go get embedded in the membrane. Okay? These are all pieces of information that are contained in this decoration. Now, I'm going to say this, and then I'm just about done. And that is that if we look at the N-linked glycoprotein, this modification that you see starts in the endoplasmic reticulum. So the modification starts in the endoplasmic reticulum, and it gets completed in the Golgi apparatus. So it starts in the endoplasmic reticulum and gets completed in the Golgi apparatus. The O-linked glycoproteins get made only in the Golgi apparatus. So the O-linked are made only in the Golgi. The N-linked start in the endoplasmic reticulum and get finished up in the Golgi apparatus. Now, these are organelles that are involved in what we call trafficking in the cell. All right? Not quite drug trafficking, but almost the same thing. They have destinations about where they're headed, where they're going to go in the cells, what's going to be their function. And those functions are determined by the structure of things that we see out here. Is that clear? Is everybody ready to finish 90 seconds early? Hey! All right. We're off and running. Yes, sir.
I just had a real quick question about that last thing.
Uh-huh.
Is this part made in the ER and this part's made in the Golgi?
It depends on the individual one. So there's quite a variety of those that you can imagine. So it's not a rule that this is one or the other.
Okay. Yeah. And you said the O-linked are only made in the Golgi?
The O-links are only made in the Golgi. Correct. Hey, Andy. I don't know. How you doing?
Be on the final.
So I had...
What's on the final?
For you, nothing. This system is acting up again. So, well, it means it was odd. When I started everything up, there was some odd noises coming out of the speakers, and I called it in, and they said it happened this morning as well, but once people got going, it went away, and it did. So be aware of it. But if it starts up, they said let them know. Okay? Yep. Hey, how you guys doing?
Good.
What's up?
I had a quick question about carbon dioxide. I'm
Yeah.
taking environmental physiology right now.
Yep.
So we went over it the other day, and I
Okay.
haven't had a chance to look in the book yet. So I'm asking you since I'm here.
That's fine.
I seem to remember you mentioning in one of your lectures that hemoglobin has a lower affinity for CO, but it will still bind to it.
I didn't say that, no.
Okay,
No.
so that's what I want to make sure, because in environmental physiology, he says it's about 200 times higher affinity for CO than for oxygen.
Yeah, it's